Okay, so these are going to be the vocabulary words that you're going to be responsible for. You're going to record those after you watch the PowerPoint, and you know our topic, which is cells. So when we look at cells, we have to figure out how we were able to view them in the first place. So when we look at the compound light microscope, that was the first microscope that was used to observe cells. So Robert Hooke was the first to observe cells in 1665, and he was able to view cork cells, which is actually from the bark of a tree. And he was able to note that these cells have a structure, which is a box-type shape, and that's how he came up with the word cell. Anton von Leeuwenhoek was the first to also observe living cells, moving cells inside a pond. And here you can see a painting of him in 1674. And Matthias Schleiden was the first to state plants are actually made up of cells in 1838. Theodor Schwann actually said the same statement in 1839, that now that animals are also made up of cells. So remember, these are things that people were visually able to look at and see plants and animals. But what was their simplest unit? When you look underneath the microscope, you can see that cells are their basic unit of structure. So again, what instrument allowed these discoveries to be made? And if you said compound microscope, you are correct. So with that, in the 1800s, we came up with a theory of cell biology. So cell theory states the following three principles. All organisms are composed of one or more cell. If you're one cell big, you're unicellular. If you're more than one cell, we call you multicellular. Cells are all made so cells are the small units of living things, which is the basic unit and organization of all organisms. And also, finally, cells can only come from other cells. So it was thought that cells could arrive by just doing combinations of different things, almost like ingredients, like in baking a cake, that if you add these certain elements, that you would get new life from it. It was called spontaneous generation. And we did that when we talked about discoveries in early science. When we talked about the scientific method, Reddy's experiment was on spontaneous generation. So we originally had thought that cells, or we weren't sure how life actually arose, so people would combine different elements and they would try to make life from that. But it was found that the only thing that could provide life were other cells. So other cells gave a rise to other cells. So we know that viruses are actually not cells because they are not able to reproduce on their own, which is a definition of a life function. So all cells are able to do the life functions that we discussed earlier. But viruses are the exception to that rule, where they are not able to reproduce on their own. They need a host cell in order to do that. When we looked at viruses, we were only able to see them when we used an electron microscope, which was in the 1940s and 50s. But all of the other cells that we view are going to be underneath a compound light microscope, and that was done in the early 1600s. So how come a cell is the size that it is? It has to do with diffusion. So cells need to be small and can't reach a certain size because if they're too large, they're not able to distribute the material. So we call this surface area to volume, and that ratio limits actually how big a cell can be. So as a cell gets larger, the volume of the cell increases at a faster rate than the volume. So if the cell radius increases, for example, 10 times, the volume will increase by 100 times. So what does that look like? If you had diffusion, if you look at a 1 by 1 centimeter, the diffusion is going to happen equally. Or as the larger the cube gets, you can actually see that the diffusion is not taking place, or the transport of materials is not happening quick enough. So we see that cells can only be a certain size. Obviously, this is a 1 by 1, that's too large. Cells are going to be much smaller than that, but cells can't be seen by the naked eye, because a cell that would be too large would not be able to be allowed to distribute all the material through it. So cell shape is going to be a lot different than what you've seen in textbooks, and we've said that in class before. Depending on the actual shape is going to determine the function. So if you look at epithelial cells, which are skin cells, they're going to have a different appearance, almost like shingles on a roof, and they're going to be there for protection. Where a nerve cell is going to have a signaling component to it, where it's going to signal from one cell to the next for communication purposes. So that particular cell is going to be very web-like, where it's going to reach out and talk to other cells through signaling process. Okay, so cells definitely appear different than the one that we see in the animal and plant cell pictures in the textbooks that we've seen. Okay, so these are two basic types of cells. Prokaryotes we talked about in class are going to be bacteria. They tend to have DNA that is not in a membrane-bound organelle called a nucleus. So we call it often free-floating DNA. And sometimes it's even called a nucleoid, right over here. So all bacteria are prokaryotes, and we find that in the fossil record, there are approximately about 4.5 billion years in Earth's history. So we see that they're the first cell to actually arise, and they're quite small in comparison to the newer cells, which is called eukaryote cells, EU meaning new and karyote meaning cell. So these organisms are going to be made up of one or more cells, and they have a nuclear-bound, membrane-bound organelle, right? So including the nucleus, right? The mitochondria are all going to have a membrane around them. So we did this in class. We compared the prokaryote versus eukaryote, and if you look at the complexity, you can definitely see the eukaryotic cells, in this case an animal cell, is much more complex than a prokaryotic cell. So the cell organelles that we've seen and taken pictures of are going to be viewed by an electron microscope. Electron microscopes are going to be much more powerful, and they're going to allow us to see very, very tiny organelles that we would not be able to see with a compound microscope. So in class, we did the animal plant cell lab, and we used a compound light microscope. You can see that you can't view very many organelles. It wasn't until we invented the electron microscope in the 40s and 50s that we were able to actually see these tiny organelles. So what are these organelles? Cytoplasm isn't considered an organelle, but it is the liquid region of where you actually find all the organelles. And notice the word organelle sounds very much like organ. So the cell organelles are going to perform life functions for the actual cell. Cytosol is sometimes also used in reference to cytoplasm. Again, it's the fluid liquid that small particles are free floating, right? So the microscope we used in the gizmos, this will also be reviewed in class. So I'll leave that for class, and we'll go over the labeling practice link in class as well. So this is just a simple explanation of what we can see. So when we look at the compound light microscope we did in class, we'll be going over all the different parts. And you can see the electron microscope is much larger. It actually takes almost the size of, goes all the way up to the ceiling, down to a desk. And here's an image where you'd actually see. So things like this, like the virus, you can see right here, this is actually the Ebola virus. You can see different organisms, and they're always black and white, and eventually you can add color as you view them on the electron microscope. You can come in and color them. You can see the viewing part right here. We use electron beams to come down in a vacuum, and that allows you to see small organelles. And here you can actually see the mitochondria right here, and this looks like rough endoplasmic reticulum. You can see the little ribosomes out there. So it was not until we invented the electron microscope that we were able to see details in organelles like that, and of course viruses as well. So let's go over the cell structures. So the plasma membrane is also known as the cell membrane, and this is going to provide like the skin. So this will be like the skin, and you can see these little receptors, and that's going to receive information. And we'll spend more time on cell membranes for cell transport the next week. It's also called the fluid mosaic model. And again, it's because things are not stagnant, like this picture where they're just not moving. They actually kind of free float and move around. And again, when you saw the movie, you were able to see that. So the video clip you won't watch, but you've seen that actually in the documentary. The cell wall is actually providing structure. So remember, plants can stand upright. You see trees, you see plants, and they're able to stand upright. And that's because they have a cell wall. Cell wall is all for structure, and it's made up of a fiber called cellulose. And that fiber, you can see, it's almost like a lattice work, and it provides stability for the plant so it's able to go ahead and stand upright. There's actual pores called plasmodesmata, and that allows for material to pass through the cell wall. So you can see it here and here. So they have a cell membrane, which allows materials, and they have these little holes inside the cell wall in order for materials to pass through. So even though it's structure, it does allow for things to pass through. The nucleus is going to be the site. So usually it's always shown in the center of the cell, but that's not always the case. And it's quite large. If you had to say the room, like our classroom was a cell, a Volkswagen Bug would be the size of what the nucleus is. It's pretty big. And inside is going to be all the DNA. And here they're showing you that the DNA is all coiled up into something, looks like an X, and that's actually called a chromosome. It doesn't normally look like a chromosome. It actually looks like spaghetti in a strainer. But we'll talk about when it looks like an actual X, which is before cell division. So chromatin is not condensed, and again, that's that spaghetti in the strainer, and that's really what your DNA looks like most of the time inside the nucleus. Chromosome is a condensed part, looks very much like this X, and that happens right before cell division. And there's little proteins that kind of hold that nucleus together called histones. Nucleolus is a dense DNA region of the nucleus, and that's actually where the rRNA, so rRNA actually stands for ribosomal RNA, or you just know it as a ribosome. And just ribosome is fine. And that's actually how ribosomes are made. They're their own separate factory. So a lot of people say, well, what happened first? Do we see DNA versus RNA? And we actually find that RNA probably was the first piece of information that was transcribed. So when we think about inheritance, we always think about DNA. But when we look at early evolutionary characteristics, we find that ribosomal RNA or RNA is actually probably the first thing that was actually transcribed in genetic information. So RNA came before DNA, like the chicken and the egg. What came first? So RNA is actually first before we actually see DNA in evolution. So then we have something called endoplasmic reticulum, which is abbreviated as ER, and you can see why it's a long word to say. And really what it is, you can see, again, the structure is right around the nucleus. And that's going to be, once you have the DNA transcribing its information into mRNA, it actually leaves, right? So the DNA is stuck here, it can't leave, but little pieces have to in order to make protein. So little pieces come out of the nuclear pore, and that's called mRNA. And that's actually where ribosomes, which are stuck on the rough ER, are going ahead and manufacturing the proteins. Then what's going to happen is going to be transported and then finally into the smooth ER, which is going to make these little round vesicles, and that's going to transport through to the cell membrane. So rough ER is the ones with ribosomes attached to it. Again, this is going to be where protein synthesis actually takes place. So the ribosomes themselves are actually where proteins are going to be made. And again, the rough ER or the endoplasmic reticulum is where it's going to be packaged, right? So they make phospholipids, which is important for cell membranes, and they're going to be making proteins. All right, so they're either exported from the cell or they're going to be repairing the cell itself. So smooth ER doesn't have ribosomes. It looks smooth. And again, this is going to be making fats and cholesterol for the cell membrane. It also is found to release calcium, and that's going to be important for cardiac cells or muscle cells. We also find that it produces estrogen and testosterone, and it even can detoxify, which is found in the liver cells. So long-term abuse can cause your body to produce more smooth ER, leading to drug tolerance. So just a little interesting tidbit, so that your cells can change over time, and their ability to do things can change over time depending on the environment and the stresses that you put on them. Right, and the smooth and rough ER are interconnected. Right, you have the rough ER attached to the nucleus, and then you have your smooth ER to do all these different things. The Golgi apparatus, or also known as a Golgi complex or Golgi body, so I'm just going to move that over there, a Golgi body, is really, it looks like flattened pancakes on top of each other. And really what they're going to do is they're going to receive the packages and vesicles from the endoplasmic reticulum, and they're going to modify the new proteins. They're going to be adding things, assembling things, and lipids, and then they're going to actually deliver them. So if you had to think of anything, they're kind of like the packaging area, in a factory where they're going to package, you're going to cardboard everything up, and then they're going to go ahead and either, ship it out or transfer it around to the actual building, depending on where the material needs to go. But so they're the packaging company, if you had to think of them anyway. Vesicles are complex. They can be small, they can be large, but either way, they're going to merge with the outside of the membrane. So here you have the nucleus and the ER, and this is where the ribosomes are going to manufacture proteins. So here you can see the vesicle going to the Golgi body. It's going to be modified, and then it's going to be sent out to the cell membrane and then released. So there's different types of vesicles. Peroxisomes are actually found in plants. And they're going to detoxify and neutralize harmful chemicals that the cell makes. So a lot of times you even find vesicles that actually kind of like the waste or recycling bin inside the cells, and they take all the waste and they recycle them. That's what peroxisomes do. Glycosomes, endosomes. Endosomes are going to be important for actually taking in large material. So they'll actually take in material, they'll eat things and digest it. So ribosomes we'll spend more time on. They're also called rRNA, and they're responsible for building protein. So here you can see the ribosome. Here's two pieces. This is actually tRNA, which is kind of in the middle. And really what they do is it locks in, and then it goes ahead and it synthesizes. These are the only things that don't have a membrane. And some ribosomes are free floating inside the cytoplasm, and some are attached to the rough ER. The cytoskeleton we talked about a little bit in class. There's different types. There's microtubules or actin filaments. Some are for structure, and some serve as highways. And they crisscross throughout the cytoplasm and serve different functions. So we saw that in the video, which is kind of interesting, right, how they work. Mitochondria have a whole unit on. They're going to be creating energy, which we know as ATP. ATP stands for adenosine triphosphate, and it's a universal energy molecule within a cell. So mitochondria are going to have this process where they're going to take their, well, we'll spend more time on it, but they're going to take the glucose or amino acids that we consume in our food, and they're going to go ahead and change that to ATP, which is the energy that we use throughout all of any cellular activity. Scientists also think of this as endosymbiosis, where the bacteria actually entered inside of a cell. Chloroplasts are also found only in plant cells, but also algae. We saw that. Algae is an example of a protozoa. And they're going to use light energy with carbon dioxide gas and water that they get from their environment. And they're going to go ahead, they're going to make their own glucose and release oxygen as a waste product. So we call that autotrophic nutrition, where they're making their own food, right? They're making their own sugar, and sometimes even store it as a carbohydrate, which is a bunch of sugar stuck together. And we kind of know those carbs like in potatoes and corn and things like that. The central vacuole is really what we use to determine in a plant. It stores large amounts of liquid in a plant. It's mainly water, but it also could have enzymes, wastes, sugar. And so plants cannot move around for their source of water or food, so it allows it like a big storage area so they can survive longer periods. So the other thing to memorize is cellular organization. So unicellular is going to be one cell big, multicellular is going to be many. So here you can see it could be one cell, right? If you have many of the same type of cell, it's called a tissue. And an organ would be many different types of tissue. And then the organ system would be an example of a conglomerate of types of organs and tissues combined together. And then everything combined would be the organism itself. Okay, so we went over this, and that's it, okay? All right, so I'll see you guys on Monday, or I'll see you on Thursday.